suggest to get started. Welcome everybody, good afternoon. Welcome to the second talk, second lecture of the distinguished lecture series of the CRC 1665, Sex Diversity. I think everybody here knows me, but if not, my name is Ute Kremer and I'm part of the CRC, part of the PIs, and part of the steering committee of the CRC and also of the research training grant within the CRC. So this distinguished lecture series is aimed to bring together leading researchers in, from biomedical research, from psychology, humanities, philosophy of science, and feminist science studies, who are all relevant to the topic of the CRC, sex diversity. And it's one of the central scientific events of the CRC, and it's targeted to a broad audience, both from PhD students of the research training group, but also targeting the PIs, of course, and the university. And as I said, it's also part of the training program of the research training group. So it's especially aimed to allow early career researchers to be exposed to and especially to participate and engage in vibrant discussions with the speakers who come here to Lübeck and tell us about their research. And I should mention that this is a hybrid event, so it will be streamed, or it is streamed on WebEx, and it will also be recorded and presented later on our webpage and or YouTube. So that's why you see all these different microphones on me and on our speaker. Okay, so now it is recorded. Okay, and with this, I'm happy and honored to welcome and introduce today's speaker, Professor Melissa Heinz from the University of Cambridge. She's a professor of psychology and director of Gender Development Research Center in Cambridge. And her research focuses on the neuroendocrine or hormonal factors that influence development of gender-related behaviors. That means of gender identity or sexual orientation, but also of behaviors like play behavior in children or aggressive behavior. And one key question in her research is whether hormonal levels already before or during birth, like prenatal or perinatal hormonal levels, have an influence on brain functions and behavior later on during life. And there's a lot of evidence for this question from animal research, where you can experimentally study this question. But as you can imagine, this is quite hard to study in humans. She's one of the, I think, first and still few neuroscientists and psychologists who do research with people with variants in sex development to address this question. And this is also what we will hear about today, I think. And with this research, she has been extremely productive and very influential, with quite a few highly cited review and meta-analysis papers and original studies, and I assume some of them will be also talked about today. And because she is such an influential researcher in this field, she's also part of the scientific advisory board of our CRC, so we are very thankful for this. And we are especially thankful that you came here to us, to Lübeck, for the third time, as we heard. So already, well, not a Lübeck citizen, it takes a long time to become a Lübeck citizen, but at least you are already familiar. And thanks again for joining, and looking forward to your talk.
Yes, thank you very much for that lovely introduction, and also for the invitation to be here to talk to you about early androgen exposure and human gender development. If you want to know why men and women or girls and boys behave somewhat differently in certain aspects, you might go online and look for a book to tell you. And if you were to do that, you'd get different answers from different books. You might say, oh, The Female Brain, that looks like a good book, that'll tell me. And if you read that book by Louann Brizendine, she makes the case, you can see in the image there, that the brain is made up of computer wire. And she says we're hardwired to be different, that it's biological and it's inescapable. In contrast, you might like the book Delusions of Gender. And Cordelia Fine, who is the author of that book, would try to persuade you that it's all socially constructed, that there are no biological influences on human gender development. So what are you to think? You'd have a hard time. And if you think about it a bit before buying one of these books, you might think, let me think about some of these behaviors, like why are men more physically aggressive on average than women? Or why do girls like dolls more than they like trucks, and boys like trucks more than they like dolls? Or even something that isn't related to gender, like intelligence. Why are some people scoring higher on intelligence tests than others? And if you start to think about it in that context, you might say, well, it's probably some of each. And that's what most developmental scientists have concluded, that behaviors, including gendered behaviors, are the product of a developmental system that involves a lot of individual factors, some of them what we might call biological, some of them what we might call social or cultural, interacting over time. And that's the perspective I take and that I will try to convey to you during the talk today. So what are some of the factors in this developmental system? So some that we've identified already, not me, but lots of other researchers, are genetic factors. For instance, the X and X chromosome in most women, and an XY in most men, an X and a Y in most men. And the one I focused most of my research on, which is gonadal steroids. So these are hormones produced by the testes or the ovaries, particularly the androgen testosterone, during very early development. So before birth, prenatally, or during infancy. So we're all familiar with hormones present at puberty and thereafter. And in childhood, the gonads aren't producing much hormones, but surprisingly, before birth, the male testis is already very active in producing a lot of hormones. And probably we're all familiar with external social influences, such as parental encouragement, or the encouragement or punishment by peers for engaging or not engaging in gender-related behaviors that are expected. And teachers and even the world at large reinforce children differently if they show gender or sex-type behavior versus not doing so. And finally, children self-socialized. Once they understand that they are a girl or a boy, they want to engage in the behaviors that they see other people of their own sex engaging in. This is a little summary of what I'm going to talk about. I'm going to talk about the sex chromosomes and the gonads and the hormones they produce. I'm going to talk about several behavioral outcomes. One, gender identity, or a person's sense of self as male, female, or other. I'm going to talk about sexual orientation, which is the direction of erotic attraction. And this can be towards males, towards females, towards both, towards neither or other. And finally, a broad category that psychologists have called gender role behaviors. And this is sometimes grouped together with sexual orientation and gender identity and called gendered behavior. And includes psychological traits or behaviors that differ on average for males and females, so girls versus boys or men versus women. And most of this research comes from a particular time period in history, so the 20th century and the early 21st century, and in a particular place, and that's North America and Western Europe. We are beginning to get information from other cultural settings, but the vast majority of information is from our own. So some might be wondering, what is sex and what is gender? And this is a very hot topic right now. For the purpose of this talk, it's, I hope, a little more simple. Lots of people say, well, sex is biology, biological characteristics, particularly the sex chromosomes and the gonads. So biologically, your sex is male if you have a Y chromosome, and testes, it's female if you don't. But these can differ. So as many of you may know, people with differences of sex development, for instance, people with a genetic variant called complete androgen insensitivity syndrome, have testes and a Y chromosome, but they're female. So is it right to say they're biologically male? Maybe it is, maybe it isn't. It's a bit confusing, isn't it? There's, even in that which seems so black and white, there's a bit of confusion. And gender is usually said to be psychological or behavioral traits that differ on average for males and females. So again, this idea of gendered behavior. Some people have said if it's gendered, it's socially determined. And you may be too young to be aware of this, but in the 1980s, 1990s, early 2000s, this was what the term gender was coded to mean among psychologists, socially determined. But if you're thinking in terms of a developmental system, then nothing or very few things are really totally socially determined. So again, it's a very complicated thing. Is this a gendered thing or is this a sex thing? And when you're looking at the data I'm going to be presenting, you've usually asked people, please tick a box, male or female. And most of this research was done before you had a box that said other. And people just ticked one of those boxes. Or we might ask people to have their children be involved in research and their parents would tick a box. Or teachers might observe children in the classroom and they would just classify them based on their knowledge of the child. So is that sex or is that gender? Anyway, it's complicated, and that's why sometimes I just say sex/gender. So an overview of the talk. First, I'm going to talk about the influences of testosterone during early development on sex, gender-related behavior, and psychology. I'm going to start with the animal models because they provide powerful information that gives us hypotheses about what to expect in people, where it's harder to do proper experiments, where individuals are assigned randomly to either get hormones or not. And then I'm going to talk about the smaller amount of research on people that looks at whether there are similar effects on human development. And the framework, again, is that of a dynamic system. Nature and nurture work together and interact over time. And I want to remind you that there are many outcomes, so a lot of different sex, gender-related behaviors, and many factors, and these can differ for different outcomes. So let's say that testosterone influences one outcome, doesn't mean it influences all outcomes, okay? This is a picture of the X and the Y chromosome. And this is not trick photography. This is actually what they look like. And you can see that the X is a lot bigger and it contains a lot more genetic information than the Y. But the Y chromosome has a gene, the SRY gene, that's very important for sex development. And that's because it causes the gonads that are originally the same in all of us, be we male, female, or other, to become testes. And as I mentioned before, testes produce testosterone already before birth, and there are dramatic consequences. By week 8 of gestation, the testes are producing androgens, including testosterone. Meanwhile, the ovaries are relatively quiet, not producing much hormones. And these androgens are why you can tell if a baby is a boy or a girl at birth, because originally we have the same external genitalia, and testosterone acts through receptors on these primordial tissues to turn them into penis and scrotum. And androgens, I think, also influence the developing brain. So I'm going to try to persuade you that that might be or is the case in this talk. So let's start with the animal research. There are literally thousands of studies of non-human mammals that find that testosterone prenatally or just after birth, neonatally, influences brain development and behavior. And these effects have been called organizational because they're long-lasting. So if you think of a hormone in adulthood, you might think testosterone increases aggression in adulthood. It doesn't, by the way, but you might think it does. A lot of people think it does. But they also think if you take it away, the aggression goes away, right? That's not an organizational effect. These effects that occur early in life, at least in animals other than humans, are long-lasting. The hormone isn't there any longer, but the animal's behavior is permanently changed. In contrast, the effects in adulthood that come and go when the hormone comes and goes have been called activational. They activate the behavior, but then it can be deactivated. And the idea about the organizational influence is that they somehow change the brain permanently. So that intrigued me when I first heard about them. This is the species and the behavior that's been studied most extensively in regard to these influences, and it's the sexual behavior of the laboratory rat. And these animals, one's a female and one's a male. The males and females don't always look different. You know, the males aren't always black and white and the females aren't always all white. The researchers use different strains of animals for the males and the females because they can sex reverse their behavior by giving them hormones before birth and then in adulthood again to activate behavior. So to tell them apart, they use different strains of animals. So in a lot of studies, people have taken female rats at birth, given them an injection of a lot of testosterone, waited till they're adults, removed their ovaries, injected them with testosterone, and compared them to the behavior of other female litter mates who didn't get the testosterone early on, got the testosterone in adulthood to activate male behavior. And what they found was that the females who'd had the testosterone early on behaved like males, and the females who didn't, even though they got the testosterone in adulthood. And amongst these thousands of experiments are experiments where they do the opposite. So they castrate the males on the day of birth so they don't have their testosterone at that time. They give them testosterone in adulthood, but the males won't show any male sexual behavior because they didn't have the testosterone early on. And you see the mirror image for female sexual behavior, this lordosis posture. The females treated early on with testosterone, even if they're treated in adulthood with estrogen and progesterone that normally activates this lordosis behavior, will not show it. And the males who've had their testosterone removed will. So these are very powerful influences. So treating female rats on the day of birth with testosterone increases their later male sexual behavior and decreases their female sexual behavior. And as I just said, removing testosterone from males early on has this opposite effect. And these behaviors can be sex reversed. You can make males behave like females and vice versa. So that was sexual behavior. What about other behaviors that differ on the average for male and female rats? Well, they're influenced too. So behaviors that are more typical of male than of female animals, like playful aggression, rats and other mammals, juveniles, show playful aggression, sort of like children, wrestling and knocking one another down, that kind of thing. And it's more common in males than females. Actual physical aggression towards other males in adulthood, more common in males than in females. And both of those are also influenced by these early hormone manipulations. And the non-reproductive behaviors that have been shown to be different in that females do them more, like nest building and grooming other animals, are similarly affected by early testosterone exposure. So it's not limited to sexual behavior. And this shows a couple of juvenile rats that I once knew and studied engaging in this rough and tumble play. And in this study, we gave some of these animals testosterone on the day of birth and some placebo injections of the oil that we had dissolved the testosterone in. And the females who'd had the testosterone showed more of this play behavior than the ones who hadn't. This has been, had already been, when I did that research with the rats, had already been found to be the case in rhesus macaques. And not just by one group of researchers, but by two separate groups of researchers. If you inject pregnant female rhesus macaques with androgens, testosterone, for example, their offspring, their female offspring, will show more of this rough and tumble behavior later in life, even though the testosterone is long gone by that time. And this is data from one of these studies by Bob Goy. Again, it looks like the data for the rats. The animals whose mothers had been treated with testosterone show more of this male typical behavior. And we assume that these changes in behavior relate to changes in the brain. And their behavioral changes were seen before brain differences were described. And the people who originally saw the behavioral changes were quite reluctant to say that they were based on dramatic changes in brain structure. They posited perhaps changes in brain function. But in the 1970s, people began to report rather dramatic sex differences in the brains of rodents. And the first one that was described was called the sexually dimorphic nucleus of the preoptic area. This is part of the anterior hypothalamic puroptic region, so subcortical, not your cerebral cortex or the rats. It's larger on the average in male than in female animals. And there's another subcortical region, the anteroventral periventricular nucleus, AVPV, larger in female than in male animals. And since this time, several other dramatic structural differences have been reported in rodent brains, most of them subcortical. And these structures change in the same way as behavior changes. If you give testosterone early in development, you get a more male typical brain structure, and vice versa if you remove it. And the mechanisms, one reason people were interested in this is they wanted to look at the mechanisms within the brain that might be responsible for the behavioral changes. So they found that testosterone during early life can cause cells to die in certain brain regions, and in other brain regions, it can rescue cells from programmed cell death. So depending, again, just to let you know how complicated this is, the same thing, testosterone, will act in the opposite way in different brain regions, but the outcome is always that testosterone causes a more male-typical outcome. So in the case of SDNPOA, it rescues cells from dying. In the case of AVPV, it kills them off. Another mechanism is where do the cells grow out? Where do their axons grow out to connect to? That's influenced by the early hormone environment. And what neurochemicals do they use to communicate with one another? In some brain regions, that's influenced by the early hormone environment. And I've described mainly rodents, a little bit of rhesus monkeys, but these thousands of experiments have involved all sorts of different mammals, the most similar to us being rhesus macaques. And some of the more exotic, the one on the lower left, are hyenas, where females actually have a phallic structure, so that interested people. How does that relate to the early hormone environment? You can read all about that in the work of Steve Glickman. And guinea pigs were the animals that were first studied in regard to these influences, and people have different specific reasons for studying different specific species. And in all of these species, the same principles hold true. And these principles are that there are critical or sensitive periods when hormones exert these effects, particularly testosterone. And these periods correspond to times when testosterone is elevated in that species for males compared to females. And the times differ somewhat from one species to another, but it's always those times when the testes are normally producing a large amount of androgens, including testosterone. So androgen is the general term. Lots of hormones are androgens. Testosterone is a particularly interesting one for our purposes. So across all these species, it's when the testes are producing hormones that the critical period occurs. If you give the hormones after that period, it doesn't have the same effect. And the behaviors and brain regions that are affected are the ones that show sex differences. So there are whole areas of the brain that don't show sex differences. They don't seem to be affected by these hormone treatments. It's very specific to certain parts of the brain, and the same for behavior. So with all of that information as a foundation, people have been interested in whether these effects occur in people. And so their first question is, when is testosterone elevated in developing male humans? Because that's going to be when the critical periods are. And what human behaviors differ on the average for males and females, because those are the behaviors that might be affected. And in 1981, Smale and colleagues published data that looked at fetal and infant testosterone levels in blood samples. And they found and reported that between weeks 8 and 24, there's a surge in testosterone in male human beings. This is before birth, week 8 to 24 of gestation, peaking at about week 16 or so. And then there's a second surge just after birth, from about the 1st to the 6th month postnatal, peaking at about the 3rd month. And as you can see on the far right here, then puberty comes along in the early teenage years, when testosterone again goes up. But these two early periods are of particular interest for potential hormone effects on human development. In regard to what behavior should we look at, the majority of human behaviors and psychological traits show small to negligible sex differences. There's a minority, however, that shows moderate to large differences. And how do we measure these differences? Do I just look at it and say, that looks big to me? No, there's a metric. There's a way to quantify this. And the way that many people use, many psychologists and neuroscientists, is they take the difference between the means for two groups, in this case, boys and girls, or men and women, and they divide that difference by the standard deviation, the pooled standard deviation for the two groups. So mean group 1 minus mean group 2 divided by the standard deviation. Let's use height as an example, because we're all familiar with the idea that there's a sex difference in height. Men are taller than women, but we also know that that's not always the case for any individual man or women. Some women are taller than quite a few men. So the mean adult height in the UK is 176 centimeters. The mean adult height for women, 163. The pooled standard deviation, 6.5. And the d-value for the size of the sex difference in standard deviation units is the nice round 2.0, okay? So that's a comparator you can use. 2.0 is the sex difference in height. In the behavioral sciences, it's been suggested, mainly by a man named Jacob Cohen, that a large sex difference is a D of just 0.8, so less than half the size of the sex difference in height. So that gives you an idea that most behavioral sex differences aren't that big. A medium sex difference is 0.5, small 0.2, negligible anything smaller than that. So this graph shows you some sex differences in human behavior in terms of their d-values. And these data are based, in most cases, and where possible, on meta-analyses. So studies that combine a lot of other studies to get a stable idea of how big something is. And the biggest gender difference is that in gender identity. So most people who have a Y chromosome or who tick the box that says I'm male have a masculine gender identity, and most people who tick the box that says I'm female don't. And that's a big difference. Next, in terms of size, is sexual orientation. Again, most people who tick that box saying I'm a man are oriented primarily towards females, whereas most people who don't tick the box that say I'm a man, who instead tick the box that says I'm a woman, are not oriented towards females, but towards males. So that's the second biggest difference. And then the next bar you see is height. So that's 2.0. Childhood play behaviors are the next biggest sex differences that have been well established. And these are almost as large as the sex difference in height. So the interest in boys' toys, how interested children are, for instance, in cars and trucks and swords and guns versus dolls and other tea sets and female-type toys, those sex differences are very big. And if you use a measure that looks not only at that, but it also at things like rough and tumble play, again, the sex difference is over 2 standard deviations in size, so similar to height. Other behaviors that you might hear about or read about a lot, like physical aggression, empathy, dominance, mental rotations ability, that is the characteristic in the cognitive realm that shows the biggest sex difference. These are smaller even, so less than 1 standard deviation in size generally, but still large by standards of what's a big sex difference in human behavior. And you don't see autistic traits on this list, in case you're wondering. And that's because measures of autistic traits in the general population don't show big enough sex differences to get on the list here. I only use things that showed sex differences consistently across studies of more than half a standard deviation. So our next question is, how can we study whether hormones influence the development of these characteristics? We can't just remove the testes of children at birth or give them injections of testosterone or give their pregnant mothers ingestions of testosterone. That would be completely unethical. So people have used 2 approaches. One, they've studied people who have genetic conditions that cause changes in hormone production or response prenatally. And examples of this from people with differences of sex development, such as congenital adrenal hyperplasia or CAH. I'm going to tell you more about these 2, DSD, as we go along, so don't worry if you can't remember these words. Or complete androgen insensitivity syndrome. People have looked at gender-related behavior in these populations to try to get information about whether the early hormone environment influences human behavioral development. And a second approach has been to relate normal variability in early testosterone concentrations to later behavior or psychological traits. And it turns out that's very hard to do. It's hard to get a measure of testosterone prenatally. Those data I showed you from Smale et al. Were fetuses who had died. I mean, again, it's not ethical to just go measure testosterone in a fetus. So yeah, it's been hard, but I will talk a bit about some approaches that have been used. And in these studies, the focus has been on characteristics that show reliable sex-gender differences. So I personally have done quite a few studies looking at what toys children like. So these toys on your left are toys that boys tend to gravitate to, and the ones on the right are toys that girls tend to gravitate to. And when I first began to do this research in the late 1970s, everyone assumed that these were socially determined, that girls and boys preferred different toys because they were given different toys, and they were encouraged to play with different toys. And when I began a study to look at toy preferences in girls with congenital adrenal hyperplasia, that is what I assumed too. But I was very surprised to find that girls who had exposed to a lot of androgen prenatally showed toy preferences that were different from their unaffected female relatives. And I'm going to show you those data in a minute. But if you're thinking, but these are socially determined, you're not the only person. And there is a lot of socialization that goes on that contributes to these outcomes. But I think there's also something that happens prenatally that contributes to these outcomes as well. And that's what has interested me for many years. Why do I study these? Well, they show big sex differences, so you have something meaty to work with. The power of your study relates to not only your sample size, but also your outcome variable. How big is the effect you think you're going to see? So if something shows a bigger sex difference, you have more power. And your power in these studies is reduced because people with differences of sex development are rare. It's not very common that someone has one of these DSD. Another nice thing about toy choices is you can assess them pretty easily. So historically, if you want to assess sexual orientation or gender identity, people might not tell you the truth. You know, that what business is it of yours? How are you going to use this information? Are you going to use it in some way that might damage me? Children don't care if you know what toys they like to play with. If you bring them into the playroom and watch them play, give them access to all sorts of different toys, all you have to do is record the amount of time that they spend with each toy and then add it up. And you can have different raters look at these videotapes that we make of children playing in the playroom, or now they're not videotapes, we just use small cameras. But anyway, you get high reliability. The reliabilities are like 0.98, almost perfect. So it's very reliable and easy to do. And childhood is a period when the gonads aren't producing any hormones, so you don't have to worry about activational effects. If you're looking at sexual orientation, you're looking after puberty. We don't know that there are any activational effects of hormones, but you don't have to worry about them if you're looking at children's play behavior. And although play behavior of itself may not seem important, it relates to other behaviors that might be, like cognitive abilities. Performance on spatial tasks, for instance, later in life depends to some extent on what children have played with earlier on. So what is congenital adrenal hyperplasia, or CAH? It's an autosomal recessive genetic variant that occurs in about 1 in 14 to 18,000 births, so as I said, not common. It causes an enzymatic deficiency, typically in an enzyme called 21-hydroxylase, and you don't really have to know that. What you need to know is that it causes an inability to produce cortisol. You don't actually have to know that either, but you want to know that it causes an overproduction of androgens, including testosterone, beginning early in gestation in girls. And as a consequence, you know they've been exposed to testosterone because when they're born, their external genitalia are ambiguous. They might have an enlarged clitoris or fused labia that look a bit like a scrotum. So that's one way that they've been diagnosed. That would lead to a hormonal workup and the conclusion they have CAH. Most countries now do screening at birth, so they're identified in that way. And boys with CAH have normal to high normal androgen levels before birth. So in terms of behavior, you expect changes in girls, but not changes in boys. And the neonatal screening is more important for the boys because their external genitalia look like those of a boy. There's no hint that they have a disorder just by looking at them. Postnatally, once diagnosed, children are prescribed glucocorticoids in an attempt to normalize the hormone levels postnatally. So the question is, do females with CAH show changes in sex-typed or gender-typed behavior? Now, the first study of this that I did was with a woman named Sherry Berenbaum. We were both interested in this question. We'd read reports based on interviews, largely with the mother of girls with CAH, that they were tomboys. And this is a term that describes a group of behaviors like liking boys' toys, not liking girls' toys so much, liking to play with boys, liking to do boys' things. And we thought, I bet those mothers just are saying that because they know their daughters were born looking somewhat like boys. And we weren't the only people thinking that. Most psychologists were thinking that. So we thought, well, let's bring them into the lab and see what toys they choose to play with. Let's ask them what toys they like. So we did that, and we compared our boys and girls with CAH to unaffected relatives, sisters or brothers where possible, sometimes first cousins. And this controls for family background, family upbringing, and to some extent genetic factors other than this CAH gene. And each child was observed in a playroom on their own with a range of toys. And here's a sample playroom. I've done this study several times with several groups of children. The exact toys depends on what I can find, usually at Toys R Us or something similar. But there are cars and trucks and airplanes, so boys tend to gravitate towards those. There are dolls and tea sets and cosmetics and hairdo toys, depending on the era. And girls tend to gravitate towards them. And then there are things like puzzles and drawing that are neutral. Both boys and girls like them. And we included the neutral toys because we wanted to know if the effects were limited to the sex-type toys. This is what we found for the unaffected children, the expected sex differences. But each dot here is a child. So you can see that not every child conforms to expectations, but on the average, boys spent more time with the boys' toys, girls spent more time with the girls' toys, and there was no sex difference for the neutral toys. What about the girls with CAH? Well, here they're the yellow bar. So they, compared to the red bar, that's their unaffected female relatives, are spending more time with the boys' toys. And the boys with and without CAH don't differ. When we look at the girl-type toys, the girls, the yellow bar, compared to the red bar, the girls with CAH spend less time with the girls' toys. And the neutral toys, there are no group differences at all. So this surprised us and led to me sometimes giving lectures in that time. And I remember one undergraduate at UCLA, where I was lecturing at the time, raising his hand, and he said, but Dr. Hines, we know that these are all because parents give them these toys. So that was the general reaction that this information got at the time. And other people have also found it, however. So I think it's a robust finding. It's not just me and Sherry. Each little flag here is a lab where this has been found. And as you can see, there are a lot of different laboratories in the US and also in Western Europe and one in Japan where similar results to those that I've showed you have been found. So here, I was in the US at the time with Sherry, that's 1992, she was in Chicago. But again in 2005, both here and in the UK, and another group in Manchester in the UK has found the same thing. Group in Sweden, also using toys similar to our toy preference playroom setup, found the same result. Others have used questionnaires, and you get essentially the same result that way too. Recently, we did a meta-analysis of all these studies that have been done on toy preferences in children with CAH, and what we found was that the sex difference is very large. So in the unaffected controls across these 20 different samples of children, the preference for boys' toys and for girls' toys was over 1.5 standard deviations. And for the girls with and without CAH, the difference was over 1 standard deviation, so clearly into the realm of large effects. What about other outcomes? Sexual orientation. Well, we've looked at a sample of women with CAH, and again, I'm not the only person who's done this, but the women with CAH are less likely than their unaffected female relatives or than men with or without CAH to be not exclusively or almost exclusively heterosexual. That said, at least 50% of them report being exclusively or almost exclusively heterosexual. So it's just that they're more likely to not report that than their unaffected female relatives. Other people have looked at whether disease severity predicts this. So the most severe form of CAH is called salt-losing CAH, and 41% of women with that form in this particular study by a group in New York were not heterosexual. The less severe form, simple virilizing, the percentage was somewhat lower, but still higher than the unaffected controls, where the percentage was 5%. So again, not just one group of researchers, but several. And this is from Friesen et al. In Sweden, and they looked at different genetic mutations that cause CAH. And the null mutation, which is the most severe form, 50% of the women reported being non-heterosexual, and compared to 2% of controls. And less dramatic mutations were accordingly were smaller magnitude behavioral effects. So again, consistent with the idea it's androgen that's responsible. And again, several labs in the US and in the United Kingdom have reported similar results for sexual orientation. What about gender identification? In 2005, Dessens et al. Summarized all the clinical reports on gender identity in women with CAH. And among 250 women, XX individuals with CAH, who had been assigned and reared as girls, 4 wished to live as boys or men. You might say that's not very many, but compared to figures for the general population at that time, it's huge. So it's several hundred times as many as would be expected for XX individuals in the general population at a similar historical time. So let's say going up to 2004. So numbers of people who were saying, I may be a genetic female, I may have been raised as a girl, but I'm really a man or a boy, were very small at that time. So 1.6%. was hundreds of times as big. Also, although most girls and women with CAH don't wish to live as boys or men, they historically have reported reduced female gender identification. So this is the same sample as was reported on from my research group on sexual orientation. Again, the yellow bar differs from all the other 3 groups. So females, women with CAH were less satisfied with being female than their unaffected female relatives or than men with or without CAH. And again, none of these women wanted to live as men, but they were somewhat less satisfied with being female. And again, this has been found by lots of other research groups in the USA, in the Netherlands, in Canada, and in the United Kingdom. What about other characteristics? Well, they show smaller sex differences, and they haven't been studied quite as extensively. But on this slide, I've said yes or no, and I've said whether it's been independently replicated or not. Personally, I don't really believe a finding till it's been independently replicated. So if it's been independently replicated, you can be more confident, I think, that it's a real result. So this is the case for childhood play, gender identity, sexual orientation, and physical aggression, which is a little bit higher in women with CAH or girls with CAH than in those without. There's a meta-analysis of mental rotations outcome. Remember, that's the cognitive characteristics that shows the biggest sex difference in the cognitive realm. And no, women with CAH are not better at mental rotation or any other spatial ability, according to the meta-analysis. Empathy, one study says yes, somewhat less empathic on a questionnaire measure. Social dominance, no difference on a questionnaire measure. But again, just the one study, so no replication or non-replication. What about the opposite kind of situation, complete androgen insensitivity syndrome? So this affects XY individuals. The testes develop and they produce androgens, including testosterone, but the person's androgen receptors don't function. So the cells don't detect the androgen. It's as if it isn't there. And they're born looking like females in terms of their external genitalia because of the androgen insensitivity. It occurs in about 1 in 60,000 births, although that number could be really wrong. We just don't have that much information. Anyway, it's very rare. And as I said, they were born looking female, and they are typically assigned and reared as girls without any knowledge that things are not just like everyone else. And there's less research than for people with CAH because this is a very rare condition. But there is information that childhood play, gender identity, and sexual orientation are female typical in almost all cases in these individuals. So no differences from controls. No information on physical aggression or mental rotation performance. Empathy and social dominance appear to be affected, but there's only one study. So the conclusion there is unclear. But some evidence, again, for an influence of testosterone or androgens more broadly. I don't have a lot of time left. I'm sorry, I seem to have not talked as quickly as I might have, but I think you've gotten the general idea. And I'm going to quickly talk a little about social cognitive influences because I don't want you to think that it's all testosterone. And I've described these a little, but I'll describe them again as we go along. So what about reinforcement of toy choices by mothers and fathers? Some people said, well, these women with CAH or these girls with CAH, maybe their parents treat them differently, and that's why they behave differently. So Vicky Pastursky, who was a PhD student in my lab, decided to find out how parents would treat their children when they played with girl-typical or boy-typical toys in the laboratory. And she used the same lab setup, and this was a new sample of children, 3 to 10-year-old, with their relatives who served as controls. But in addition to looking at what toys the children chose to play with, she looked at how their parents reacted when they played with various toys. So here, again, is the toy play setup. And she coded positive reactions, things like play initiation, let's play with this, or praise, oh, you're doing that really well, affection, a little hug maybe, asking a question that helped the play to progress. Negative reactions were criticism or ridicule. Some parents would even say things like, you look silly playing with that. Or they might refuse to play with a child. ignore the play of the child, or suggest an alternative toy. So here are the children playing on their own, and you see the yellow bar is again different from the red bar. So we saw that the girls with CAH are playing more with the boys' toys, and less with the girls' toys, just like in the prior study, but new kids and new toys. The data for play when they're with their mother or with their father, many studies don't have fathers because it's hard to get fathers to come in. It used to be, it's less so now, but it used to be that moms were often at home taking care of the kids, so they'd bring the kids in, but dads were busy at work. But we managed to get fathers and mothers, and we tested them separately. So mom with the kid, dad with the kid, and we counterbalanced who went first, mom or dad. And when the kids played with their parents, their behavior was pretty much identical to what they did on their own. So I'm not going to show you all those images. They look the same. But what about what the parents did? Well, parents tend to be positive towards their children when they play. So there were lots of positive responses. And the girls with CAH in particular got positive responses from their mothers when they played with girls' toys. They got more positive responses than their unaffected sisters did. And my idea about why this is happening, which is against the idea that the parents are causing this, is that their parents really want them to play like other girls do, but they tend not to. But if they're in the playroom and there are some toys that are for girls, they will try to get their daughter to play with those. And the fathers do the same thing, even though they're tested separately. So they give the girls with CAH more reinforcement, positive reinforcement for playing with those girls' toys. Parents are almost never negative in the lab when they come in and play with their children. But the people who sometimes got negative reactions to their play were unaffected boys, and that happened when they played with girls' toys. And this was true for mothers and fathers, sadly. So, yeah. But most parents didn't do that. So another thing we looked at is self-socialization in girls with CAH. So I think there's some role for parental treatment, but it's in the opposite direction of the testosterone effect. What about the girls themselves? So we labeled objects as either girls' toys or boys' toys, or for girls or for boys. And we showed children videotapes of adults picking certain objects consistently. So I'll tell you a little more in a minute. So typically if you tell children in general, this is for girls, let's say an apple, or more realistically, a toy horse. This is for girls, and this is for boys, like a toy cow, or a red balloon and a purple balloon. One's for girls, one's for boys. Then the children like the one they've been told is for their sex. And so we did this with balloons and xylophones. Green balloons are for boys, silver balloons are for girls, or vice versa for half the children, and the same with xylophones. And the girls with CAH, again, the yellow bar, were different again from everybody else. They were less responsive to this information. So although most children will want something if they're told it's for their own sex, girls with CAH are less likely to do so. They're not unlikely to do so, so 45% of the time they want it too, but the other kids are over 50%, up to 60, 65%. And these are actually the behavioral preferences. I thought I had them in a different order. So if you say, what do you like? You see that they're less likely to like those things. If you watch them in the playroom with the green balloons and the silver balloons and the xylophones, they're less likely to play with those items too. So more powerful type of evidence. And in the modeling paradigm, we showed the children videotapes of people choosing neutral objects. So 4 women would come in and say, there'd be a pen and a pencil on the desk. I like the pen. And then 4 men would say, I like the pencil. And again, all of this was counterbalanced. And there were apples and bananas, all sorts of different things that are neutral. And again, the girls with CAH were different from everyone else. They were less likely to respond to this video presentation by liking the things that were chosen by people of their own sex. So I think that both parental socialization and self-socialization are altered in the girls with CAH. So the point I want to make here is it's not just that testosterone has found some bit of their brain that likes a doll or doesn't like a doll. It's a complex developmental system where they and their parents, everybody's playing a role in the development of this outcome that may have been initiated in part from testosterone before birth. I want to remind you that there's a little postnatal surge in testosterone too, and I've been studying that recently because you can study, still the studies of clinical populations are studies of clinical populations. These people have other things going on. It's not just that they have a lot of androgen prenatally, they also take treatment every day. They have surgery sometime to feminize their external genitalia. There's a lot else going on. And so it would be useful to know if within people in general, concentrations of testosterone early in life relate to later behavior. My attempts to use measures that some people think reflect early prenatal testosterone exposure have been fruitless. So for instance, measuring testosterone in amniotic fluid and relating it to later play behavior, I find no relationship. I also find no relationship of those testosterone measures to autistic traits. I find no elevation of autistic traits or autism in girls with CAH, and evidence is accumulating from other people as well that autism and autistic traits don't bear much, if any, relationship to prenatal or early hormone exposure. And they're the only paper that's looked at whether amniotic fluid testosterone relates to fetal blood testosterone has found no relationship between these two. So I don't think that is a good approach or a useful, reliable approach. Some people use finger ratios. You've perhaps heard about this, that the ratio of the length of the second to the fourth digit is different on average in males and females. Again, I don't find that to be reliable in terms of predicting behavioral outcomes. And I think if you read the literature on that, you find a few studies that find what you might predict if androgen were playing a role, but you also find a lot of studies that don't. So anyway, I thought maybe this postnatal surge will work because it's much more accessible. You can actually get samples from infants and relate those to later behavior. So I'm quickly going to summarize 2 studies that did that. One, we looked at testosterone in urine samples and related that to later play behavior. And one, we didn't measure fetal or children's digit ratios, but we measured their genitalia, which we know are influenced strongly by early testosterone exposure. So these urine samples were taken, I'll show you in this graph, from day 7 postnatal to month 6. We saw elevated testosterone in boys compared to girls. The shape of that curve looks very much like the one from blood samples from Smale in 1981. But these are urine samples. And I did this in collaboration with a group in Finland who took the urine samples and did the hormone assays. And we used a questionnaire that parents completed about their children's behavior when they were later in about 18 months old. And it shows a large gender difference, this questionnaire. And we've used it with people with CAH. Girls with CAH score higher on this questionnaire. Male typical scores get a higher score. And this is what we found for our correlation. So in boys, R equals 0.46, which is a large correlation. 0.5 for a correlation is large, 0.3 is moderate. In girls, the correlation wasn't significant. And this study of the genitalia was based on data from a longitudinal study that people at Addenbrooke's Hospital were doing to look at normal development in relation to environmental endocrine disruptors, which is an entirely different topic, but they had measures of genital development, because that is one of the things that could be influenced by these environmental disruptors. And so we used the measures that they had taken. And they'd measured something called anogenital distance at birth, which is influenced by prenatal testosterone. Other studies have shown this to be the case. And then they measured physical parameters, body length, penile length, at 3, 12, 18, and 24 months of age. And later on in life, we had the parents complete that same questionnaire about gender type play behavior. And what we found was that both anogenital distance at birth, which reflects prenatal hormone exposure, and growth of the penis from birth to 3 months postnatal, so during that postnatal surge in testosterone, but not growth later on after that surge, predicts later play behavior. So this could be this or the urine samples or even saliva samples where you measure hormones could be a good way to look at hormone effects in the normal population. Yes, and so I am out of time, and I'm just going to skip over some other things. Pink, for instance, which is another interesting topic. And I also want to remind you that different outcomes are influenced by different factors. If you look at how far on the spectrum of the average female to the average male a woman is, or a girl is, if she has CAH, you'll find that she is about 60% of the way from male to female as a group. I'm not saying about an individual. I shouldn't say she. I should say they as a group. The mean scores, and this is also exemplified in the meta-analytic data I showed you, the play behavior is about 60% of the way from the average female to the average male. In contrast, gender identity and sexual orientation seem to be influenced to a much smaller degree. So there are other things we clearly don't understand that contribute to those outcomes, or we can debate why the effect on those outcomes is smaller. But I just want you to remember that if you hear, for instance, that play behavior might be influenced by early testosterone exposure, doesn't mean everything else is. It doesn't mean aggression is, doesn't mean sexual orientation is necessarily, or to the same extent. You have to look at all these things individually. So I hope I've convinced you, maybe not, we can talk about it, that testosterone plays a role in human development, and that its influence on sex or gender-typical play, sexual orientation, gender identity are fairly well established. It appears to influence some behaviors more than others and may not influence all sex or gender-related behaviors. We don't know yet. People may be unique in having gender identity. We never say gender when we're talking about the behavior of rats, for instance. So other species we don't think have this sense of self as male, female, or other. And testosterone early in life may influence gender identity, and alterations in gender identity or processes related to it, like self-socialization of gender, could increase or reduce influences of testosterone on brain development and behavior. And I think this is an interesting thing to look at. I also think that the early postnatal period, that some people call mini-puberty, is an accessible window where people could study how different types of influences, testosterone, for instance, and socialization, interact. And it's a period when the brain is continuing to develop rapidly. You can get repeated samples of testosterone from typically developing children. And you could look at testosterone and how it interacts with other factors. And I just want to conclude by thanking funders, the United States Public Health Service, the Wellcome Trust in the UK, and the European Economic and Social Research Council, or the UK Economic and Social Research Council, and lots of collaborators. So, and thank you for your attention.